Whole genome sequencing at birth. What research can and should be done with a baby's genome? The hashtag for the event is Genomics Conversation, and this discussion has been made possible thanks to the generous support of Genomics England. So at the event tonight, I'm hoping that the panel will address and then we'll get to discuss some of these questions, such as how significant might the benefits to research be if researchers could make use of this genomic data from newborns, and how do we know that that's a sort of robust evaluation, not just hype? How and by whom will it be decided that particular individuals, organizations, companies can access genomic data from newborns for research purposes? And what will those researchers be allowed to do with that data? And perhaps just as importantly, what won't they be allowed to do with it? And if parents decide that they want their baby to be part of this program and have their whole genome sequenced, will they be able to place conditions on the use of that baby's data? Will they be able to opt out altogether from research, or cherry pick, say, well, yes, I'm happy for it to be used for cancer research, but perhaps not for something like obesity? And how can the public make its voice heard on these really important matters?
Well, good evening, everyone, and thank you very much, Sarah, for inviting me to be part of this. So I'm going to try and do is just put this this issue in the context of day-to-day life in the neonatal unit, in the neonatal intensive care unit, and then talk through some of the issues that Sarah has already alluded to. I want to pay tribute to Genomics England, because I think this is really a landmark, innovative way of approaching this issue, which is going to have resonance across the world. And a particular aspect for me as a clinician and a researcher is that, of course, by embedding it in the care of the NHS, we're ensuring that there will be equity of access to all patients, but also there will be a heed, a mindfulness, a heed to the duty of care, not only to the individual patient, but also to that patient's family and to broader society. And if we get, let's say we do go ahead and we are in a position where we are offering whole newborn sequencing to all babies born in this country, stop and think about how important those duties of care and candor are. Because, for example, we've all seen some of the problems that have played out through direct-to-consumer whole genome sequencing. So I think this is a really landmark aspect. And the other landmark aspect is that the research program in relation to the newborn is being co-designed with families and the public and professional organizations. And that too adds a particular set of layers of value and relevance and trust to this program. And I may come back to that as we go through this talk. So neonatal intensive care units, neonatal units, there are about 200 neonatal units in NHS units in the UK. Pretty much every baby who requires over and above normal newborn care is admitted to one of these 200 neonatal units in the UK. And we admit collectively about 100,000 babies to these units each year. Now, our patients range from full-term babies with, for example, complex multi-organ support needs to extremely preterm babies who may be born up to 18 weeks before their due date, and everything in between. And about 2,000 babies die in their first month each year in the UK. In a neonatal unit such as the one I work in, we would expect to have, on average, roughly about one baby dying each week. So this is quite a toll, not only on families, but also on staff who are regularly having to support and counsel families through these illnesses. Now, the genomics program and whole newborn sequencing primarily makes reference to rare diseases, and rare diseases affect around 6% of the UK population, and roughly about 10 children are born each day with a treatable rare disease. There are about 7,000 rare diseases that we know of, and of these, about 80% have got a genetic component. The conditions are disabling, they are expensive, and they usually bring great tragedy to the families. 1/3 of children with a rare disease die before their 5th birthday. And the majority of patients who undergo standard testing do not receive a diagnosis. So there's a huge unknown and uncertainty around these conditions. And for us as clinicians, it's important to note that rare diseases often masquerade as common diseases. So a very common masquerading feature is for a baby to admit it who seems to have a serious infection. And it can often be a few days, and sometimes it can be much longer than that before the penny drops and there's realization that actually this isn't an infection and this may be a rare disease. And by that point, the baby may well have suffered, may have died, or may have suffered permanent damage. So early diagnosis, being alert and instigating the right tests early on is really, really important. But whole genome sequencing is not just about rare diseases, and this is where we move into the future. And this is where I should mention the growing prevalence of, for example, the chronic non-communicable diseases, currently responsible for a huge number of deaths. Onset is now earlier and earlier, and the rise in the population prevalence of individuals who are affected by one of these diseases and are living with lifelong disability is growing. But prevention, if we're going to get smart about prevention for these common diseases, these common non-communicable diseases, we really need to get smarter about prevention. And of course, whole genome sequencing offers future possibility of better prevention through better understanding of gene environment, gene medication, and gene-gene interactions, to name just 3 areas. So this is the excitement of the future, because rare diseases are rare and common diseases are common. And through whole newborn sequencing done carefully, we have the opportunity to affect and influence and improve not only the way in which we manage rare diseases, but also the way in which we think about common diseases. Now, the 100,000 Genomes project produced some very, very important research information, but a lot of the studies that accompanied it have also been hugely valuable. So, for example, an NHS study suggested that as many as 20% of selected neonatal intensive care patients may have a genetic disorder. A paper published in the New England Journal of Medicine, the lead author was Sir Mark Caulfield, who led a large amount of this work. I'll give you 2 examples here. One of a baby boy who died at 4 months, but the diagnosis from this study enabled the testing and treating of a younger brother. And another more indirect example is of a man in his 60s, whose father and brother and uncle had all died from kidney failure. He himself had had 2 kidney transplants. He had an affected daughter, but after a genetic cause was identified, his granddaughter was tested and found to be negative. What a relief for the child concerned, the family, and also how important to recognize that child and that family was spared the huge anxiety of not knowing what the future held, and also spared the surveillance and the investigations at regular intervals that were the only thing that healthcare could offer until that diagnosis was reached. So in this pilot study, 25% of proband, that's the initial presenting patient, were able to receive a diagnosis. 25% of them were able to receive immediate, actionable changes in their care, hugely beneficial. Now, at the moment, as Sarah has said, the current newborn blood spot screening programme only screens for 9 diseases. They're listed here. It's a blood spot programme. We know that there are at least about over 600 fully penetrable disorders presenting early in childhood. What scope through research for actually improving our diagnostic capabilities and therefore bringing benefit to patients? However, there are lots of buts. And here is a wonderful quote from Sir Neil Gray, former programme director of the UK National Screening Committee. He said, all screening, please read the words carefully, He said, all screening programs do harm. Some do good as well. And of these, some do more good than harm at a reasonable cost. Very, very wise words indeed, which we should heed as we go through this. So, the impact of whole genome sequencing on patient care actually hinges upon doing the research very carefully and very, very wisely and very well. What's here already in terms of the impact of whole genome sequencing is disease management. If you make a diagnosis, you can really change disease management. You can introduce a new treatment, you can change diet, you can introduce a replacement therapy. You can even cure some diseases with, for example, organ transplantation or stem cell transplantation, bone marrow transplantation. And what's often forgotten about is you can redirect care. What's fast coming in terms of the impact of whole genome sequencing research is better reproductive choices. The ability of a family to make choices based on evidence of what diseases they may be carrying as a family, or may have arisen as a de novo, a new mutation in an affected child, which may not have any implications for future siblings. So better reproductive choices, better genetic counselling, and also better counselling and better information for the rest of the family. For example, if a baby is found through whole genome sequencing to be at risk of a hyperlipidemic disorder that may affect other members of the family, testing other members of the family may be really important for their lifelong health consequences. But the research questions are, how do you tell them about that? Who do you tell? When do you tell? And then into the further future, we are going to be bringing nearer personalized medicine, personalized care plans, and targeted therapies. They're coming fast and furiously, but we want them to come in a way that is well managed and is ethical. So my final slides, the uncertainties that we need to resolve through a very carefully conducted research program. What are the false positive rates? False positives bring additional costs, bring anxiety, bring stress. What are the false negative rates? These bring delay in diagnosis and inappropriate reassurance. How can we consent appropriately for wider research use? And who should be involved in the consent process? If we are talking about a baby's genome, when should we disclose results? To whom should we disclose those results? When does the child gain autonomy over their own decision making? When should they be told about a condition that might not affect them at all, or might affect them only in later life? Who should have access to the genomic data? How can that access be properly governed? What are the ethics of commercialization? And how can we ensure that any future research, secondary and tertiary research, actually brings public benefit? And this is particularly an issue when the primary reason is commercial benefit. Surely the public or the patient group who have contributed their genomic data should also be able to benefit. So lots and lots and lots of reasons. And finally, as a neonatologist who runs a research program and who runs a large national neonatal clinical database, we have recently been asking the views of young people about the data their data that are held in the database. These data are held with their parents' agreement and their parents' knowledge. But at what point should we tell the young people themselves about the data? How should we tell this to them? How can we ensure that they know? We've conducted quite a lot of research in this area, and one very interesting result that has emerged is that young people, not surprisingly, have very, very different views from those of their parents. So it's extremely important that we ask the young people themselves, and we think carefully about whether it's the parental views or the young person's views that might be relevant to the particular question we're addressing. And we need to bear in mind here as well that genomic information will be relevant not only to an individual, but also probably to their health across the rest of their lives and also into the next generation. And again, we have data showing that families are usually very generous and very altruistic. We've conducted genomic studies where parents have been very willing for their baby's DNA to be stored in an appropriately governance repository and for that DNA to be made available to other researchers. Again, they want to be reassured that this is appropriately governed. So I will end there with saying that this isn't about putting any genies back in bottles, but it is about cautious advance. It's about honesty, transparency, respect for individual use, respect for the autonomy of the individual. And the individual in this case is the baby, who is not as yet, when you obtain that first genomic information, the baby is not able to exercise that autonomy. And finally, I think it's about trust in science.
Our next speaker this evening is Dr. Imran Kausar, and Imran is the UK Vice President and General Manager at Novartis Gene Therapies and responsible for developing gene-based therapies for rare conditions.
So I have been asked to talk about industry's perspective on the Newborn Genomes Programme and generally how such programs could benefit the acceleration and development of medicines, particularly for young children. It's no surprise that the pharmaceutical industry, Novartis Gene Therapies in particular, are interested in creating valuable therapies that can improve, enhance, and perhaps ultimately cure disease in adults and in children. But it can't be ignored that actually, if you look at the overall span of drug development, a lot of it's based on adults and diseases of adulthood. And up until recent regulatory changes, actually the vast majority of licensed medicines that had come out were focused for adults and adult diseases. There was some overlap where a disease might present in children, so for example, asthma, and those medicines could be used for children as well. But until regulations changed to increase the ease with which studies could be run in children and the regulatory aspects, actually became very difficult to run studies in children. However, since those changes, we've seen an increase in the number of licensed medicines that are for children and aimed at childhood conditions. But this still leaves us with a very large gap. And looking at the most recent data, we can see that although there are more drugs available now, licensed drugs available to treat children and diseases of childhood, there are actually hardly any for the treatment of neonates, the group that were just discussed by the last speaker. So we have a gap, and the potential that newborn genomics has in allowing us to bridge that gap cannot be underestimated. Because although many of us don't see neonates in our day-to-day life, apart from those like Neena and the countless others that work in neonatal units up and down the country, actually there are a number of conditions that can affect children as young as that, but also children in general, which require a further and more in-depth look to see how industry and research in general can better serve the children and the needs of those children. And of course, one area where that can happen is in genetic medicine, but also in rare diseases. So the Newborn Genomics Programme offers a superb opportunity for industry to be able to better understand a lot about the basic mechanisms that are impacting childhood diseases, the stage at which they come on, the proteins that are affected, and allow us as an industry to be able to understand which part of the disease process we should be interacting with to try and understand and mitigate and ultimately cure the disease. But that's not as simple as it sounds. And even if we reflect on adult drug development, we can see that identifying a particular target, understanding the role of the target in the disease, understanding the role of genomics in that particular disease is a highly complicated affair. But at least in adults, we have the opportunity to speak to adults, gain consent from adults, and a large number of them to be able to understand disease across the different types of disease as it manifests. With children, that's a lot more complicated, in particular with very young children, that's extremely difficult. So projects like the Newborn Genomes Project give us a remarkable step forward in being able to understand many things. And let me just give you some examples on how they relate to drug development and the pharmaceutical industry. So as an industry, our interest in making medicines means that we need to have a really strong and in-depth understanding of the disease that we're trying to affect. And although it may seem pretty straightforward that a disease can be caused by a genetic mutation, actually the reality is that it's far more complicated. The gene and our book of life is not a simple book to read. There are numerous variations. There's bits of code that are copied and repeated, and sometimes they're backwards and forwards. And there are numerous variations which all need to be understood in terms of, are they harmful? Do they have a role in altering the disease? Or is it simply a case that they do nothing? But that needs to be understood. And the best way to understand that is to be able to have access to that, but also have access to another important thing, which is the patient's data over a period of time. A strong asset of the program is that, as well as the genomic code, the actual DNA that could be harvested through genome sequencing, actually how the patient is performing. There are other biomarkers, aspects related to performance in other areas, on the heart, on the brain, etc., all provide valuable insights into how a disease is progressing, when it manifests, how it manifests, and the impact on that child. A single gene mutation can have a number of outcomes in children depending on the age at which you pick it up. But the fundamental understanding of the disease biology and the process of disease is extremely important for drug makers to be able to understand the right target to follow and the right outcome to understand. In many severe diseases, or those which are strongly life-limiting, a simple outcome might be a prolongation of life. But there are many conditions where actually children will survive, often into adulthood, but in a profoundly disabled state or a state where there's significant functional impairment. And in those instances, it's critical to understand at which point do we consider an improvement as an improvement? And also, is there an opportunity for us to perhaps make the patient worse? It is possible through working with genomics to give too much of a particular protein, and that could have some negative effects as well. So the fundamental understanding of disease is critically important, and it's very difficult for us to do as an industry. A huge benefit, and why we're at Novartis Gene Therapies, but I'm sure my industry counterparts are also very supportive of this project, is because of the access to data which we can have through a very transparent and in a manner which allows consent to have been drawn up front, but then for it to go back. And that's something which exists in the current 100,000 Genomes Programme, which has been remarkable for Novartis, but also will be very important for any future project aimed at newborns, because consent is extremely important. And as Neena mentioned, you know, it may be that there's a future point where children who were enrolled into programs may no longer wish to provide their consent. However, in understanding the dynamics of the disease over a longer period of time, that's important. But the key thing is we have the opportunity to be able to do that and to explain the findings of our results and hopefully the benefit of having some treatments. Newborn genome sequencing is complicated, and it should be complicated. And this level of debate is very important so that we can understand the nuances of the debate, but also the implications of what we're about to do. Neena mentioned about popping the genie back in the bottle. And of course, we would say, well, science needs to have value to society. And with regards to neonates and young children or diseases that specifically impact young children, as an industry, but certainly as a society, I think there's a lot more that we can do and should be doing to better understand their diseases and to find treatments and to find therapies that work for them and at least improving their outcomes and hopefully one day providing a cure. So I think with that said, the newborn genome sequencing offers a tremendous opportunity to get an in-depth understanding of disease, which should have the outcome of better drug development, swifter drug development, personalized drug development, in order to tackle some of the most profound diseases that affect the youngest and most vulnerable in our society. And the industry is committed to working in a transparent manner with Genomics England and other bodies around the world to make sure that the regulations exist to be able to do this in a transparent manner where consent and the families and the child's needs are always placed at the forefront of any decision making.
Our next speaker this evening is Michelle Mackie, and Michelle is head of Ipsos MORI's Qualitative Research and Engagement Centre.
What I'm going to be doing is sharing some insights from Ipsos MORI's catalogue of research and engagement with the public over the years, which relate to the use of health data, including genomic data, in research. So just a note on that. This, as you know, is quite a new area. So many of the studies that we have done with the public since we started the conversation around 2015 relate to health data sharing more generally, more widely. However, we have used examples of whole genome sequencing and genomic data across those projects. They were sort of futuristic at the time, if you like. So just a bit about sort of where this insight comes from. We started having conversations with the public through deliberative and dialogue methods, which is a sort of more deep and in-depth means of engaging and asking these questions compared to sort of surveys and even focus groups back in 2015 for Welcome. And you can see across the years, we've sort of dipped in and out of the conversation, covering consent procedures with the HRA and HTA, sort of future of data-driven technologies with the AMS, a dialogue for Genomics England itself, which covered the future of genomic medicine and included whole genome sequencing in that, and right up until today, as well as current projects that we will be able to speak about shortly. So what do we know across all of these projects in relation to what the public are excited about, worried about, and so on? So with regards to aspirations, sort of thinking back to sort of some of the points that Neena made in her presentation, participants in these studies have recognised the value of using genomic data. They're surprised that sequencing technology exists, but they do recognise those benefits to individuals, to dependents, and also to wider society and the NHS as well. However, they have concerns around genetic data being viewed as potentially the most private of all sort of health data and sensitive, and also the most potentially valuable. So this could shine light on areas of life which have so far been quite mysterious, such as infertility, allergies, a predisposition to obesity, for example. Is this too intrusive? So this is a question that the public sort of ask themselves when they're talking about this. And there, of course, are very many unknowns. So, and this could potentially have dangerous repercussions for society. So they talk about profiling and designer babies and, you know, sort of future unknown and potentially quite damaging applications. But after deliberation, where we spend quite a lot of time sort of explaining and taking participants through this in deliberation, people do appear to be quite happy with this uncertainty and accept it and trade this off against the sort of complete confidentiality for potential benefits. You can see here, there's a sort of quote from somebody who took part in the dialogue back in 2017 around sort of the future sort of unknowns as well. So there are a number of red lines that we've observed across these studies, and this again relates to the wider use of health data in research. Genetic engineering being one of them, the link to sort of designer babies and using technology to sort of to change people in terms of cosmetics and that kind of thing is completely unacceptable. Using genome data and wider health data for surveillance, so things like taxation, immigration, and crime and justice. Any kind of administrative or political uses is a complete no-go, as are predictive insurance and targeting and marketing. So obviously we have to tell the public that commercial organisations are involved in this research. If they weren't there, then we wouldn't be able to make drugs and make treatments for people in the future. And this does cause a bit of alarm to some people, and there tends to be less support for commercial companies having access to data, notably because of the fear that altruistic donations of data will enable private sector profiting. So sort of selling off data, worries about sort of data being used for reasons which were not agreed initially, but also a sort of strange assumption that data security and handling won't be up to the scratch of the NHS, for example. I don't think this is helped by some of the sort of news stories that we've heard over the sort of past 10 years in relation to sort of data loss and issues that have happened, because people remember those. There's also a big expectation, speaking to some of the points from Neena's presentation too, around the fact that any use of these data would have to result in public good. And there's been lots of sort of work on what that means, what public good and what public benefit actually means. There was a piece commissioned by the National Data Guardian last year, or the year before even. And people with rare diseases tend to be more clued up about the risks and more cautious about ability to re-identify in the data. Some that we've spoken to in our dialogue have had experience of commercial companies exploiting their health status and marketing products to them. So a slide that we presented as part of the Welcome work back in 2015-16, which still applies to every single piece of work that we do since, are really as sort of drivers of acceptability. So in summary, it's all about with the public, why, who, what, and how. And you can see here that genomic data, genetic data, under the sort of category of what does sometimes appear to be less acceptable because of that sort of more sensitive, more risk of re-identification because of the level of the data, so that's just sort of something to keep in mind. But it really is about, is there a clear benefit? Who's going to be involved? Who's going to access data? What it's going to be used for, and how it's going to be stored. And I'll just finish off with some sort of ideas for sort of future public engagement, which I think are really important. And Neena already touched upon some of these, and I'm sure lots of people are thinking about them too. So whose consent are we really talking about here? Genomics England commissioned a whole genome sequencing dialogue last year that Ipsos didn't carry out. But this found that for the use of data in research to be, this was an expectation that use of data in research must be accompanied with an effective and dynamic consent process, which is one which requires ongoing engagement between individuals and users and custodians of data, and that the child should consent when of age. So that was a voiced expectation from that piece of work. And the second point is, can genomic data ever be depersonalized? So much of the reassurance that the public have when we talk to them about the use of their health data comes from knowing that health data can be depersonalized, even though it's kind of not completely impossible. Re-identification is hard and illegal, but obviously that risk sort of increases given genomic data is completely individual.
I turn now to our final speaker this evening, who is Professor Mike Parker. And Mike is Chair of Genomic England's Independent Advisory Ethics Committee, and he is Professor of Bioethics at the University of Oxford.
One of the things I've noticed, I suppose, worth saying at the beginning, one of the things I've noticed about this initiative is often in the media, it's described mistakenly as the sort of national newborn screening programme as opposed to the Newborn Genomes Programme. And I think that the actual name is a much better one because it captures, it makes it clear that this is a research programme and research and evaluation programme, and it situates what is a really ambitious and exciting initiative in the same kind of space as some of the other of the UK's major internationally important research initiatives, such as the 100,000 Genomes Project. And I think that's right. And perhaps in some of its other closest cousins, maybe some of the birth cohorts that the UK is famous for. So the Millennium Cohort Study, the 1958 birth cohort study, USBAC, and so on. And clearly it's not a cohort study of that kind. There are a number of differences, and I'll come back to those later. One obvious one is the scale, and another one is the close link with the NHS. But we can come back to that more later. So I'm really delighted to be here this evening because I think it's very important as part of this. The point made about public trust by others. It's really important to critically reflect on, to identify and critically reflect on the ethical dimensions of this programme, and we need to ask ourselves the question, well, what would be required for this to be judged by reasonable people, to have been undertaken in an ethical way, a way that's conducive to reasonable, to well-founded public trust and confidence? I think that's an important kind of set of questions to ask. So I'm going to come in a minute to sort of talk about the ethics of doing this, some of the ethical challenges, but I also think one of the things we don't do enough in ethics is to think about the ethics of not doing things. We tend to jump straight into the ethics of doing things. I just thought, there are morally significant reasons for a program of this kind to exist, and some of them we've heard about already. But I wanted to sort of disaggregate some of that a little bit. So the first, morally significant benefit of this program is research relevant to improving understanding, identification, diagnosis, and treatment of rare conditions, and we've heard a lot of reasons why that's important in the talks previously. But I think there are 2 or 3 other sort of research elements of this that are important. So one is second is the research and evaluation of the idea of creating a resource, a lifelong resource, whereby genomic data produced at birth can be combined with other information, be a lifelong resource of benefit to the person throughout their life. So there's an important element of this, which is research and evaluation of that idea too. To what extent is this a lifelong resource that could be of value to people? And I think there's a third research element here too, which is potentially research on the extent to which preventative approaches to medicine informed by this kind of analysis and other data early in life might avoid disease or lead to more effective earlier interventions for disease later in life. So I think these are sort of 3 scientific games which are really important. But also I would want to argue that there should be an embedded ethics research program, ethics and social science research program built into this too. And my understanding is that would be the case, because I think it's really important that we learn as we go about the ethics of this, and that's done in a systematic way, complementary to the engagement work, but sort of systematic academic work around the ethical issues. So I think these are 3 or 4 reasons why it's ethically important to do this program, because we're going to learn a lot from it, and there are potentially huge benefits. But now I sort of want to move on to talk about what some of the ethical challenges are and what some of the things we might need to think about are along the way. One important thing, I think, is to think about should there be feedback to participants, and if so, what sort of feedback should there be? And it's an important question to ask because at one time, not so long ago, it used to be standard practice and expectation to make a clear distinction between medical research and clinical practice. And there are a whole range of reasons for that, but it was taken to imply that researchers should not be feeding back clinical information to research participants. But the world's changed since then, and that's no longer sustainable. We've seen the move from models like UK Biobank, which have a no feedback policy, towards 100,000 genome projects and so on. Participants now expect this as part of the deal. So I think a position of no feedback is no longer sustainable. So that leads to the question, well, what should be fed back? And a whole range of questions here, and it's partly a medical question about what's meaningful, what can make a difference, and it's partly a question about what people want, and what participants would like to see. So that this adds another research program, I think, to this, which is, to what extent, an evaluation of the feedback that this program is going to provide to parents and so on, might this offer the basis, or might take the form of constitute a carefully evaluated large pilot for something that might become a national screening program in the future. So I think this is an important research element too, and it makes sense. I don't think this research would be sustainable or people would sign up if there were no feedback. So let's do a research project around the feedback, and what can we learn? What can the NHS learn about that from that? So I think that's the range of questions about feedback that we need to think carefully about. Related to that, I think there are ethical questions about the importance of establishing and meeting reasonable expectations. So if we tell participants, parents, that they will get some information back about their child's genome, what's the timescale? What can they reasonably expect? And how will we communicate them if things change with them if things change? So I think creating reasonable expectations among participants is a really important ethical consideration. And then we come to the question about deciding, whether to participate or not, and what's required for a parent's decision to enter their child into this study. What's required for that to be a valid decision? And it's inevitably going to be broad consent, because the parent at the time can't be informed about all possible research that will be done on this data. It's inevitably going to be to some extent open. But I don't think that means that it's invalid consent. I think we make decisions that are open-ended all the time. The key thing is that, genuinely state-of-the-art efforts are made to clearly convey what the purpose of the study is, the ways in which information will be stored and accessed, how decisions about access and how decisions about research will be made. And that's combined with transparency, with the avoidance of any kind of deception, that the decision is voluntary, and that people have a right to withdraw. So I think there's a whole lot of questions about consent and decision making. And we can talk about those later, perhaps. But I think one of the most important questions that arises along the way, and I think Neena was the first to raise this, is, well, what should young people's input into that be? And clearly they can't have an input as neonates. But there is an important question about how children and young people are going to be empowered to have greater amounts of decision making about the uses of their data and the conduct of the study as they get older, and ultimately to take over control and decision making about the uses of their data. What's that going to look like? How's that journey going to be managed, and how can it be done in a way that's ethical? And I personally, building on the point that was made earlier, I would strongly advise the setting up of a children and young people's advisory group to feed into this process throughout the life of it. I think that's absolutely essential. And then we move on to the question about the uses of data, and we've had some discussion of that earlier. I think the idea of a trusted research environment is really important, and that's, you know, the way in which things are increasingly being done. And I think this is a good thing. Genomics England really established the model for that. And this is the idea that researchers come to the data rather than the data going to researchers. And that means that the activities that are undertaken on the data could be audited, that we can be clear that the researchers are doing what they say they're going to be doing, and there can be careful controls in that space. There are all sorts of practical ethical considerations about the creation, the nature of trusted research environments and the licensing of them, but those are kind of issues that maybe we'll come on to at some other point. But of course, the fact that the data is in a trusted research environment doesn't answer the question of what should be done with the data, what kind of research is acceptable, and it doesn't answer the question about who should be allowed to do research on data. And of course, that raises the questions that Michelle raised about commercial companies and so on. And research in this space is not viable without commercial partners. So how should we think about that, and how should we manage that in a way that is conducive to appropriate trust and confidence in what's happening? So that requires careful thought too. And I think then there are sort of two further questions, perhaps. One is, given that consent is going to be broad and that people are going to be required to place their trust in the creation of this data set and research on it, I think we need to also ask ourselves the question of, what do we need to do to ensure that those who participate in this study over the life of their life, as it were, are not going to be significantly harmed or discriminated against as a result of their participation. And that's important for its own right, of course, in its own right, but it's also crucial to address that question for the initiative to be viable and sustainable, and for public trust and the trust of the participants to continue. We want this to be done in a way which is successful and doesn't lead to lots of people withdrawing their consent because they're very concerned about what's happening. So how do we manage that in a way that means that people are going to feel safe participating? And then I think, I mean, I was going to stop there. I think I'd just say one more thing about it. I think it raises important questions about innovation in healthcare. So I think I once heard a talk by Matt Fennick, where he distinguished two different models of innovation. So one is the model that is in Silicon Valley, which is, you develop something, you release it, and then you innovate over time. You learn from the way it works in the real world. And then there's the other model at the other end of the spectrum, which is the model of the clinical trial, where you have a very robust, long, expensive process, and you don't release medicines into the world, as it were, until you're really confident that they're safe. And it strikes me that in this sort of space, when you're thinking about these kind of initiatives, we need something that's somewhere between those two things. We need, and I think this program has the potential of creating a model for ethical innovation that's both flexible and dynamic, but also careful and conducive to public trust and confidence.
And I will just now quickly introduce Dr. Richard Scott, Rich, who is the Chief Medical Officer at Genomics England.
I think the panelists and you, Sarah, have set out where we are with the programme so very clearly. I'm not going to add a great deal at this stage. I think to emphasise that, as you said, Sarah, we're at the beginning of this programme, and to be clear about timeline, we have more than a year before we start to recruit people into the programme. We're at this co-design phase where events like this evening, engagement with the public in ways that you've heard about, we have before, and also through an NHS steering group that we've established with with experts to really co-design this programme and dig into some of the questions that we've touched on today about the research, but also many other elements. I think it's also the way you set out those three areas of exploration is really useful to keep in mind around the early diagnosis for treatments available today, around research into novel healthcare findings, for example, of improved diagnostics, earlier diagnostics and treatments that wouldn't be otherwise available, and then this concept of the longitudinal life course potential of genome data. And while that middle area of exploration is the one with research in the name, as you've heard from all of the panelists today, research is broad across not just how the program's conceived as a research pilot embedded in the NHS, but under research ethics, but also in those different areas there are research questions. And I think the sorts of question that Mike has teased out around, for example, this needing research not just in the ways perhaps our minds often jump to it, in those very particular scientific questions about the potential of a new treatment, around the questions about whether or not whole genome sequencing does or doesn't add value in particular settings or in general in terms of identifying conditions that are treatable very early, but also in how a program like this might run in research, in the ethics, in terms of, and also the practical implementation. And so many of those questions are really tightly linked to each other. So what we're really keen through events like today and through the work that we're doing already and in the coming months is to have that dialogue with participants who will be, we're building on the foundations that Mike's talked through about the concept of a trusted research environment where data is accessed under terms that the participants in our programs are happy with, and having that dialogue with the public, with potential participants and with researchers to explore exactly what uses people want their data to be put to, so that we can answer the questions they want. So we're really keen to engage in that through tonight, through other events, and we're really looking forward to hearing the discussion and being part of that.
And so the question is, would you expect children to reconsent when they're adults, and who would be responsible for that? I think it's a sort of a theme that's come up a lot in terms of the importance of that, and it's something that Neena raised, but I think we'll perhaps go to Mike first with this, to talk about this, this type of consent, and whether it's whether we call it dynamic or what we call it, but I think that would be helpful, Mike.
So although I am the chair of the Ethics Advisory Committee for Genomics England, I'm actually on the board. I'm not speaking for Genomics England this evening, so Rich will probably be our best place to answer the practical questions. But there is clearly an important set of ethical questions here, which are really important. And I said that decision making. I would say not just when they become an adult. I would say that over the course of a childhood's life and growing experience and maturity, they ought to be more and more involved in this process, as insofar as that's possible. Clearly, there will be children for whom that's not possible, and they may well be some of the people that the initiative is most interested. So for people with developmental disorders, for example, may find it more difficult to get involved in decision making. But nonetheless, I think the emphasis should be on where possible, and as much as possible, young people should be involved in decision making at an early stage. And of course, at some point, they should take over that role completely if that's possible. I suppose the two things to say is, firstly, yes, I think that should happen. Secondly, clearly, when they consent or decide not to consent, there will be things that will have happened before they get to that age that they won't be able to have a decision about. So we need to remember that parents will be making some decisions that are not reversible. For example, a child's in a study at the age of 2, and their data is included in that analysis and then published. They won't be able to get out of that retrospectively, of course, but they will be able to. So parents need to bear in mind their responsibility when they're doing that. Nonetheless, I think, yeah, later in life, children should be able to decide for themselves whether they want to be involved. And there are models like ALSPAC and other cohort studies have been along this journey.
And Michelle, do you want to come on that from the research and the work that you're aware of?
Yeah, I mean, it's really interesting, the sort of comparison with the cohort study, because we carry out some of those at Ipsos MORI, and we've had to think about, when the child becomes of an age to consent and how to engage them in a project that they're part of, that their parents initially signed them up for. I mean, the Child of the New Century, the Millennium Cohort Study, is a fabulous sort of current example of that. They're turning 24 now, and some of them are becoming harder to engage because they're going off and doing different things, and maybe going to research. Being part of a research study isn't sort of the most exciting thing in their lives anymore. I think that the sort of the piece that I've covered previously, as I said before, we did find that the real sort of expectations from the public is that there should be a sort of ongoing dialogue with regards to consent, not just a sort of blanket consent, and if and when it's possible for the child to become involved in that.
And Neena, you raised this issue of children and parents not necessarily thinking the same thing. Do you want to come in on this?
Yes, very much. Thank you, Sarah. So my research group has done some work on this. I can't speak about genomics data, but I can speak about clinical data because I lead a large neonatal research database with clinical data, and we have done work speaking to families, to parents, and to young people who were former patients themselves and who have now grown up. And what I would strongly, the clearest message that comes out is that, as always in research, we should not go into this with any assumptions at all. So what the young people have told us about their data. So we went into this saying, well, we would like to ask for your consent. We've got, your parents have said it's fine for your data to be in our database. We would now like to, you've grown up, we would now like to ask you whether it's okay for your data to be in our database. And they said to us, essentially, we are fed up with being asked things which ought to be happening anyway. What we'd really like, is to be able to access information at a time of our choosing, but we don't want to be bombarded with things endlessly. And then they said something else very interesting, which was that, and remember, this is not about genomics data, it's about clinical data, but they said, we've grown up in a world that's completely different from our parents. We share our data all the time on social media platforms. Every time we go online, it's not a big thing to us. And we know that companies are using our data for all sorts of commercial reasons. What we'd really like to be assured about is that the health service is using our data for things that are really important to improve health. We'd like to be able to get more information if we want it, but we don't want it shoved in our faces. And then we don't want this need for consent to be shoved in our faces. That's what they said. And then they said something, they gave us a very, very interesting suggestion. And this has happened very recently, so we haven't had a chance to follow up on this, but a very interesting suggestion. They said, when you're telling our parents of a baby in a neonatal unit about the database and about the research that's done with the database, why don't you also give parents a leaflet that they can give to their child at a point when they think their child is sufficiently developmentally, cognitively ready for it? Now, I thought that was the most brilliant suggestion. Because how do you, if you've got 1, 2, 3, 4, 10 million people in a database, how do you actually practically go back to them and ask them for their consent? Not possible. But you do want to inform them. And the final thing I'd say is that what they really emphasized, both parents and young people, was what they really value over and above consent is feeling part of the process and being informed. So I thought fascinating feedback from our parents and young people groups.
That's really interesting, Neena. Thanks for sharing that latest data. I think it sort of made a really good point there. And Imran, before I go to Rich to say what Genomics England are thinking about this. You did emphasise in your presentation, the importance of consent to Novartis. Is there something that from an industry point of view you'd like to see here?
So I think I'd say for a start, the interest in consent, I'm sure, isn't just of interest to Novartis, but across the industry and indeed anybody that's performing human research. But I think perhaps what's relevant here is the fact that the nature and the expectations of consent have changed over time. And I think what people might have accepted many years ago with clinical research, they're perhaps less willing to today. And when it comes to their children, and in particular with genomic-based research, I think there's a different set of expectations, and those expectations need to be met in a way that fulfills their needs and can evolve. And so I think from an industry perspective, as often we are one of the bodies that will reach out to an organization like Genomics England to say, we have a problem or an interest or a question, and can you help us with this? We would fully expect that during our due diligence, we will have had a full set of understanding of what is the extent to which the data that an organization has can be used, and can we go back? Was the consent process to a high enough level? Does it conform to international requirements and regulatory requirements, and does it fulfill the company's requirements for consent for a given population? So I think, although we don't ever see individualized data and certainly never identifiable data, it's always at a cohort level, at a large group level. The concept of consent is extremely important, because fair research can't be conducted if the participants aren't fully aware of how their data are being used. However, I think in saying all that, I think there is also a lot of not misunderstanding, but perceptions around what consent means, what people understand from it. Some are very happy to undertake consent very rapidly, and others perhaps want a more detailed view. And of course, we're guided by ethics committees as well, which take a strong view. And more, and particularly in the world of research into rare disease and younger children, patient associations will also have a view on consent materials. We're recently about to run a trial where we're looking at the consent that we're providing as a company to undertake the trial. And we've been working with the patient groups to make sure that the language that we use is understandable to families, particularly where it's complicated or there may be implications. So I think the notion of consent is an evolving process, but it's certainly of deep interest to pharmaceutical companies and other researchers, particularly where it comes to rare and young children.
And finally, come to you then, Rich. Have you got Genomics England got any ideas of how this may be done, or how you may tackle this issue?
Yeah, so we am absolutely in agreement with the other panelists. This is, the expectation is that as people come of age and are able to make those decisions themselves, they need to be in a position to understand how their data might be used, what their choices are. So at the moment, for example, we are working with our participants from the 100,000 Genomes Project to go through exactly this exercise, and we're doing this together with the NHS, and we see it as our responsibility as Genomics England to make sure that happens, even if we're not the people having all of the conversations. I think it will evolve a lot over the coming years, and by the time a newborn cohort come of age that's recruited in the next couple of years, things will have changed a lot. And I think, as we've heard, that really what the challenge here is for people to be engaged in the program, to understand, earlier than they come of age, if that's appropriate, in discussions, how their data is being used, so that they're part of the program. And so that rather than us thinking about this as, a piece of paper or some sort of box that needs to be signed, it's actually about people having the sense of sort of engagement in the program as a whole, and control and contentment about how their data is being used.
And so this is Sophie Peat from Genetic Alliance UK, and she says research into treatments for rare conditions often pull data from across the world due to small patient populations. So has the use of whole genome sequencing data in international research for treatments for rare conditions been considered? And do the public feel differently about data being used outside of the UK? I think that perhaps we go to Rich first with this about how researchers can currently access data from the 100,000 Genomes Project. Then perhaps we'll go then to Michelle for her thoughts on this from the public attitudes.
Thanks. So in terms of our current arrangement, what we try to do is get the balance between that the security one going through a trusted research environment, where essentially the model, as you've heard, is of researchers visiting data and carrying out research under the expectations of the participants within within that trusted research environment, as opposed to approaches that perhaps other studies have taken historically, where data is more broadly available. So the researchers accessing the data are international. People don't have to be physically in the UK to access it. They have to go through the right processes, be accredited, and be considered trustworthy by our participant-led access review committee to do that. So that's one part of it. We also are working with other sort of big studies internationally and within the UK to work out how within the expectations of our participants, we're able to make data available in a way that can help make some of those discoveries. And that's something which is still very much a sort of work in progress to make that work really well. People talk about sort of a nirvana of so-called data federation, where data stays in one place but can support research across different data sets. That's something that we're actively sort of exploring, but it's something which is hard to deliver, but is very much part of the future as we see it.
Michelle?
Yeah, so as you can imagine, the idea of data sort of going outside of our own domestic ground worries people when we talk to the public about this, just naturally because of the sort of uncertainty around whether sort of safeguards and data storage and security and access procedures are equivalent to those that we have here, and whether the sort of law would protect the rights of individuals. I think at the same time, the public that we've spoke to recognise that like particularly pharmaceutical companies are global companies. So, you know, when you're talking about developing drugs, then you can't necessarily always assure people that will be done in Cambridge or wherever. So I think there is a bit of a tension there. I think it's one that people would need to understand, and it would be, you know, it would need to be explained very well to participants. And as well, totally agree with the sort of concept of a trusted research environment, and we're starting to have those conversations in current public engagement that we're doing for the London Health Data Strategy Programme, which is the NHS part of within London, and also for Our Future Health, which is a new programme, a UKRI programme, where they're interested in engaging the public in conversations about trusted research environments. So the conversation with the public has moved on to sort of, how do you feel about your data being used, to like, how do you want it stored and accessed?
Thanks, Michelle. Imran, so Novartis is a global company, as most pharmaceutical companies are, let's face it. Do you have any thoughts on this question?
I think perhaps there's a misconception if people feel that their individualized identifiable data is traveling across the world to end up in some unknown place in some unknown institute's hands. And I think that naturally would cause concern. The reality is that even research that's generated today using conventional methods, which are widely accepted, those data, when they're aggregated and turned into averages and medians and modes, actually there's no way to identify an individual from that, and those data are shared. And in the questioner's context of rare diseases, it's actually really important that they're shared, because otherwise we don't have enough patients to be able to get an understanding. So I think perhaps this comes back to a fuller explanation of precisely what it means when your data is shared, and to introduce the concept that it's never an individual's data or an individual's genetic code. It's an average or an aggregate of those data that provides a that provides a better, more complete understanding. And again, as an industry, this is something that we have to do with regulators, and people no doubt will be familiar with the recent therapies that have come around for COVID and how data has been shared all across the world with tremendous success to be able to help aid decision making and improve therapeutic options. So I think a lot of concerns can be at least better understood if there's a better explanation of how specifically and precisely how data can be used, but also using examples of how this could be a beneficial thing and not necessarily something which is concerning or could cause any risk to anyone. These are all great questions, and the fact that questions like these come up mean that I think people still have concerns, and data leaks and things happen, and naturally people, this is valuable data because it's an expression of yourself, and you could feel as if you're being robbed or something is being stolen from you if this isn't handled correctly. The trusted research environment is an excellent way to be able to assuage some of those concerns. But I think for the vast majority of cases, these will be aggregated data that you can never identify anyone from them, but the concerns need to be addressed comprehensively.
So we'll move on to an anonymous attendee. So whole genome sequencing is a wonderful thing, a wonderful technology, and a proven use for children with unexplained symptoms and signs. It's much harder to interpret the significance of any variation in the genetic sequence in healthy babies. So as whole genome sequencing is now so quick, why not wait until a child is symptomatic before embarking on whole genome sequencing? Unless, of course, there are clinical benefits to making a diagnosis pre-symptomatically, e.g. with SMA, spinal muscular atrophy. So I don't know who wants to go on that one first. I think perhaps we'll go to you, Neena, first, about the importance of doing this early.
Sure, thank you very much. Yes, it's absolutely invaluable to make a diagnosis before the onset of symptoms. For very many diseases, once symptoms have started, or one is very rapidly running out of time. And with many of the serious rare diseases that are being addressed through this program, a baby can be irreparably damaged while you're waiting for a diagnosis to be obtained. So absolutely crucial to get an early diagnosis, which is one of the reasons why I mentioned the case history in my presentation, which your questioner may not have been present for, which was the story of a baby boy who died, whose whole genome was sequenced, who did have a diagnosis made, but who died. However, because the diagnosis was made, his brother was able to have treatment implemented, instituted, started before that child was damaged by the disease. And that's the case for rare diseases. It's also likely in the future to be increasingly the case for common diseases, which are really in the research arena. And I'll just give you one example, very quick example, and that is the problem of obesity. Now, I'm not suggesting that obesity is a genetic disorder, but clearly there will be genetic components. This is where gene interactions with the environment and with other exposures come in. But obesity is best prevented. It's very difficult to treat obesity. Many of the chronic conditions that are so common and are absolutely debilitating our societies today are best managed through prevention, not through treatment. So it's a generic point. Prevention is always better than treatment, and early diagnosis is pretty much always a very good thing if you have something that you can do about it, if it's an actionable disorder.
Do any of the other speakers or Rich wants to come in on this one? Rich?
I think Neena's sort of framed it very well. I think it's about recognizing that as well as the 9 conditions that we currently include in the Hill prick test in the UK, there are a really quite large number of other conditions where if one misses that window of opportunity, where identifying a diagnosis early before usual diagnostic processes are able to, it's very hard to explain to families why we haven't tried to look for those conditions. The research pilot is about asking exactly this question about, which conditions are we able to do that effectively in? How do we do that in practice, and what value does it bring, and what are the challenges and pitfalls? So it's absolutely the right question, and it's about us doing it right and recognizing it's different from diagnosis, but brings very important value, that it's very hard to sit in front of families in clinic and explain why one's not trying to do this. It goes back, I guess, to Mike's point around sort of recognizing the danger of inaction.
I don't know if there any other speaker wants to come out here, but I suppose one thought that occurs to me with this, so there's a sort of a practical issue here, that when people have a baby, they're very much part of the healthcare scheme, and so it provides the opportunity where I suppose if you're asking them to do it at a later point, that could give rise to problems. I want to sort of skip down my list of questions now to a sort of related question, which is to Imran, but I think others will want to come in on this, is can you explain a bit more why whole genome sequencing data from newborns is needed for drug development, and why this can't be achieved by whole genome sequencing of adults who are volunteering by providing their samples. So Imran, do you want to go with that one first, and then we'll come back to others, because we've sort of covered this slightly from Neena.
Yeah.
But
I think that there are a few very good reasons why adults with the same disease couldn't act as proxies for children with the same disease. I think the first is that there's some diseases which only happen in very young children, and as a consequence of the disease, they may not survive into adulthood. And so to understand those diseases, for example, some cancers, you can only use children or neonates with those particular cancers. So that's a very clear reason where adults can't be used to extrapolate data or to understand a neonatal environment. The second issue is that adults who can participate in research are survivors. And so therefore there are a large number of children with conditions, both genetic and not always rare, that don't make it into adulthood, or at least by the time they are into adulthood, they may have significant functional impairment. And so by taking their genomics at that particular point will give us a snapshot of the genome. So it might give us some indication of the genetic fault that may be causative or have some role in the disease. But actually doing it at the stage of a newborn gives us a much better picture of what impact does the genomic sequence and then the creation of the protein and all the downstream effects have at a much earlier age. And by the time of adulthood, that's not there. The third thing is safety. So medicines are all assessed on their risk versus their benefits. And the way that adults metabolize medicines is different to the way that particularly young children metabolize medicines, and we can't always use the same data sets to make those assumptions. And so understanding newborn genomes gives us a better view of the genetic profiling, but also what's happening in the child at a very young age. As I said in my brief talk earlier, it's not only the genetics or the genomics that are of interest to researchers, it's also the other biodata that also go along with it. So the heart rate, the blood pressure, blood test results, etc. And so those provide a much clearer view of the overall impact of any given disease on a child. So those are some of the reasons why adult data can't always be extrapolated. Although interestingly, it was the way things were done in drug development for many, many years. You just took what happened in an adult and you worked it backwards. And many clinicians on the call are listening in will be familiar with the days of off-label prescribing, where you just took a drug at a dose that was designed for adults and used them in children because there weren't any data from children. And that's generally not considered to be best practice anymore. And regulators have created incentives for companies to make sure that they are asked to do research in children where it's appropriate and ethical. So there are a number of reasons why adults with a particular condition aren't a useful proxy for children.
So the next one is, with reference to genomic data allowing better reproductive choices, how can we avoid discriminating against people with genetic disabilities? So, so perhaps we'll go to you first with that one, Mike, and then we'll perhaps come into Michelle to see if that came up in the work that she's done. That's those sorts of questions.
Yeah, I mean, I think, well, there are a number of things to say about this. Obviously, there are a number of ways of thinking about the potential impact of the uses of genetic testing in reproductive medicine on groups, people with disabilities. And so there's one argument that's often made is what's called the expressivist argument. So that simply by making these choices available, we're saying something about the value that that society places on the lives of disabled people, and that clearly it's extremely important that that message is not one that comes across from this. I think one obvious way of addressing this issue is to involve people with disabilities and groups representing people with disabilities in the process, and that's I would assume that's what. So when we're talking about involving young people and engagement activities, that attention will be paid to that. So I think that's the key thing that can be done here, and. The other thing is, of course, this is a standard problem or challenge in clinical genetics or reproductive medicine more broadly, which is, when parents are being provided with information about their pregnancy, that's done in a way which is genuinely informative and is supportive and is one that, the questions about the implications of this for their possible future child's life are discussed in a way which don't imply that a decision one way or the other is what's required or accepted. So acceptable. So it's essentially a lot of questions to do with the broader politics of this, questions about involvement, and questions to do with communication with patients and parents at the time.
I think most of the insight that we have is focused on sort of use for research rather than the sort of programme itself.
The next question that we have is, how will a balance be struck between industry's need to generate profit and respecting the public's altruism in donating this data? Will the public be expected to pay for drugs and tests that are developed off the back of this data? What would be the impact of NHS privatisation on this? I think that we might park the NHS privatisation bit, because that's not yet happened to my knowledge, unless I've missed something in the news today. But we'll perhaps go to Imran first on this, as it's a question for industry. We'll let you tackle the difficult thing first, and then we'll let the others come in.
I think it's a great question, and indeed it's not a new question. Individuals or government-funded grant organizations have participated in research that industry have used for decades, and so the notion of a public-private partnership is not new in this context. But I think ultimately the question is about fairness. What is a fair price for innovation? What is a fair price for taking the risk of creating medicines? The vast majority of therapies and medicines will fail, and that's a risk that companies take on. And so I think in answering the question of fairness and pricing of medicines, which is a very important question, particularly when it comes to rare diseases where prices can often be extremely high and development costs can also be very high as well, I think it's a very important question. And in the UK, at least in England, there's a body that kind of checks for value of medicines. Of course, that's nice. And they perform cost effectiveness analyses to ensure that the NHS is getting value for value for money in the medicines that they're acquiring. However, as there's a greater request upon society to participate in research, particularly innovative research like this, it's not difficult to understand that people may feel that there is a greater onus upon companies to offer something that reflects that commitment from a country or commitment from state-funded research, and that should be reflected in the price as well. I don't think there's one answer that anyone can give that can fully solve this problem, but I think the notion of fairness in pricing of medicines is very, very important, and it's an active and ongoing dialogue which changes as patient expectations and health system expectations change as well.
Now, Neena, you raised this issue in your presentation about whether people who have donated their genomic data in this way should be able to have a share of the profits down the track. So if you want to go next, and then we'll go to Mike.
Yes, it wasn't. I didn't think I said profit, but I think I said something about benefiting from it. And yes, to follow on from Imran's very reflective comments, the facts of the matter are, I think, evidence that there is a lot of work to do in this space. So at present, over 95% of the medicines that we use in neonatal intensive care are used off-label or off-license to this day. Now, we had the US Food and Drug Administration pediatric ruling way back in 1998. We had the European Union medicine, the EMA's pediatric ruling in 2007. Despite this, these were incentivization schemes to promote the development, to incentivize the development of medicines for babies and for children. Despite this goodwill, we have only in the entire history of newborn medicine had one medicine that was ever developed for newborn use. Let me repeat that. There has only been one medicine that has been developed for newborn use. Every other medicine has been a hand-me-down medicine, and 95% of those medicines used in neonatal intensive care are still used off-label or off-license. So Imran's absolutely right that this is a rapidly moving field. There's a lot of discussion that needs to be done. I don't think the world has quite got it right yet. And there does need, this is because this program, the Newborn Genomics Programme, is directly targeting newborns. It's a huge opportunity to actually work closely with the medicines industries to try and improve where we are. This is something, again, that evolves over the years. We have to work together to get this right. But the notion of a fair price, I completely agree with Imran, because the pharmaceutical world does contribute a lot in the way of development costs. They do take a lot of risks. But on the other hand, I see it from the point of view of patients and the NHS, which also contributes a huge amount, also takes a certain amount of risk. And this should be a partnership. And whether we've got the balance of that partnership right is something that's clearly going to evolve over the years as well.
Mike.
Yeah, I think this is a really important question. And actually, you know, the relationship, the way, the sort of social contract around data and healthcare in a data-driven sort of health system, you know, with research as part of that, is a really important question that we need to take seriously as a society. I think that's actually crucial. So don't want to. I think that's a big question. I sort of had three things to say really. The first is I think it's really important to disaggregate the different ways, the different types of commercial or private companies that might be involved in medical research. So there are clearly going to be some companies that are drug companies. There'll be others that are technology partners, you know, sequencers, etc. And there'll be others who, you know, I don't know, design software or whatever it happens to be. But I think, you know, at the moment when we talk about commercial involvement in medicine, I think the public, and I guess I would have thought the same thing too, and that's until previously, you think pharma companies, but actually it's much broader, a much wider range of kind of types of involvement than that. And I think that's actually quite important, because it comes to Michelle's point about, well, what are in those red circles and what are in the green circles, and I think people may have different views about those things. I think the second point is, and this may link to what with Michelle would say, but you know, I don't think we're necessarily as worried about profit. It's profiteering we're worried about. You know, of course companies need to make money. The question is how much, and you know, what's the appropriate way of thinking about in the context of this kind of partnership. So I think, you know, the point that was just made by Neena, you know, but this is about what can we expect, what can we reasonably expect of companies that are being enabled to do work on NHS data. You know, what's a socially responsible but nonetheless profitable company? What does that look like? I think is an interesting question that we need to think carefully about. And then coming back to the sort of social contract idea, you know, we don't. This is not about privatizing healthcare. This is about sort of, you know, not patient facing, but sort of research and so on. You know, we don't expect the NHS to make its own computers or make its own paper. You know, we expect the NHS to interact with companies at in various ways. The question is, what's an appropriate way, and you know, is that well managed and in the public interest? And that comes to this question again about the sort of social contract. And I really think it's a very important question for us to address as a society.
The next one that I'm going to go for is one for Rich, and that's, will all parents be given access to genomic counselling ahead of deciding to consent on behalf of their child? Will the child receive genomic counselling later in life?
So absolutely, families will be provided with the right sort of information to give them an informed choice. One of the things that we're talking about is exactly who would provide that and exactly what sorts of format. Most likely, different formats for different sorts of people. Not, the opportunity to ask a person questions face to face, but also increasingly people tell us that, for example, accessing information asynchronously by videos, etc., complemented by discussions. So absolutely, whether it's genomic counsellors specifically, whether it's midwives, whether it's other trained individuals, essentially the important thing is that these people who are considering joining a program or their baby joining a program have access to the right information and are able to ask the right questions. I guess one real advantage to a program like this is that having a baby doesn't come as a something without warning, and therefore actually there's a period, a busy period, and one where healthcare professionals are busy interacting with people, but a period to allow people to think about their choices, to really explore that. So that's one of the things that we'll be exploring during the research pilot, is exactly how we best do it, how we provide the right information, but also how we do that in the context of a busy NHS, how you can practically make sure that there are the right number of people to have those conversations.
I've got a couple of questions I'm going to sort of lump together for Neena now. One is from an anonymous person saying, for some metabolic conditions, tandem mass spectrometry is much more accurate, fewer false positives, false negatives, and whole genome sequencing. Have you considered the impact on NHS specialist clinics and on families if lots of children are actually well, get referred to these clinics? How will this affect or dilute the services for children who are really affected? And then another one from S. Batter, who would like a bit more information from you, Neena, about what are the false positives and false negatives rates with whole genome sequencing.
Thanks, Sarah. Actually, I think this is more a question for Rich, because this is really part of the research programme. We don't know. As Rich said, there are a large number of potential diagnoses that could be made using whole genome sequencing. But until the research is done, we don't know what the false positives and false negatives are. And so we don't know what this wider requirement for resources or what the wider impact on the NHS is going to be. But Rich may want to come in there with more thoughts about how the programme is going to tackle this.
Yeah, please come in then, Rich.
Yeah, so we're working through at the moment in detail how you'd go about selecting what disorders to look at. But we know from work that we've done over the last few years and published by ourselves and others that there are a very large number of conditions which either there is no other way of identifying a diagnosis easily. For example, tandem mass spec just can't pick up. Or whole genome sequencing along with other follow-up tests is a really good way in to make a diagnosis. So we think there are a really good number of conditions where it could potentially add value. We think that the list is probably more than 200 conditions. As Neena said, a really important part of this research pilot is to dig into the details of exactly how one selects those conditions, exactly how one runs the computer algorithms that examine the genome, exactly how laboratory scientists use that data to make decisions potentially alongside other data. For example, it's possible to use the mass spec data that we use to look for those many of those 9 conditions already alongside a genome to make really good informed decisions and so forth. So working through all of that is the work of the programme. And that false positive, false negative rate will depend on those decisions, but very clearly in the context of newborn screening, the false positive rate, the way we tune those all of those decisions and those algorithms needs to be very low for it to be something that's workable. And that's something that we can see as is a very important part of how we design the programme.
So we've got one question from Annabelle Slater and an anonymous one. I'm going to mash them together. So Annabelle wants to know is, will young people have rights over the use of their genomic data only when they reach the current legal age of consent for their own medical treatment? Or will a different legal limit apply? I suppose, it'd be interesting to know if there would be a hard, age once you've turned, 13 or whatever it is, arbitrary date, that would be the decision time. And then from someone who's anonymous is, in regard to children, if parents consent, have you thought about the potential consequences for the child in regards to insurance and careers when they become adults? And obviously some of those things are things that Michelle flagged as potential, sort of issues for people.
I just think potentially there's an opportunity here for this project, if that's the right word, this programme, to be innovative and about its involvement of children and young people. So there is a question about what children and young people's legal rights are over their data. But I would like to see us sort of go a bit beyond that and say, well, how, what's best practice in this space? What can be done to include young people as much as possible, and to find ways of hearing young people's voices, even if they're not necessarily the participants. Can, as I said earlier, can a young person's advisory group be set up, for example, to test as a sort of sounding board for some of these things? So that's sort of background, but I would like to see sort of greater involvement of young people in decisions about their data as they get older. So it's not so much for me about rights. Obviously, there is a sort of rights question. It's about sort of responsibilities of this initiative to do the right thing for young people. So that was that one, and then the other question was about insurance, and I think. I was on a Nuffield working group a few years ago about data, and that one of the most important findings in that report, recommendations, which I think found a lot of resonance, was the idea of sanctions for misuse of data. Such as trying to identify people and so on. So I think, those kind of protections are very important. But I also think, work needs to be done to ensure that. That this doesn't, as I say, discriminate or harm the young people as they get older. So an insurance would be a worry. Employment might be another. So that needs to careful thought.
I would be really interested in sort of more sort of in-depth public engagement with young people, either that sits alongside the programme or that informs it at various stages. I don't think it's unreasonable to suggest that these views could really be powerful in terms of shaping the consent process in particular, because otherwise we're talking to parents and it's not enough.
And a question that's come in for you sort of specifically, actually, Michelle, is from Imran Rafi, sorry if I've mispronounced it. Did you pick up on any cultural or religious concerns that may exacerbate diversity issues in non-participation?
Yeah, it's a really, really interesting question. And it's also one that lots of big data programs are thinking about with regards to getting volunteers on board, keeping people engaged and communicating about how data are used. I think we've found nuance. We've found some sort of nuance within the data that we've collected from our conversations with the public that suggests that some different communities, some ethnic groups are more cautious about the use of their data. There's some sort of inferences around sort of fear of surveillance and sort of intrusion that come through in some of those conversations. I don't think we've completely nailed it though in terms of like doing specific work with specific groups, because most of the work that I presented earlier has been a sort of a more general sort of cross-cut of the population, either in England or across the UK. But I think that is a really, really good area to think about and to investigate further.
Emily Tullock wants to know about how do you share research so that it can be understood by pupils at school who are studying biology, or for their teachers who are trying to inspire a new generation of geneticists or genomic counsellors. So I don't know who wants to take this one. It's
shall I take that one?
Yep.
Because this is a question dear to my heart. So this is such an important issue, and it's really important for all of us who sometimes, when we're speaking to our colleagues, speak in shorthand and jargon, but really have to be able to communicate much more better than that. So one of the things that we do in my research group is I ask everyone when they're writing their papers to make sure that they also write a summary that is going to be comprehensible to anyone who is not a specialist. And that's not necessarily included in the publication, but it's a very, very good discipline to help clinicians and scientists to kind of force them to practice communicating what can sometimes be extremely complex concepts in words that are comprehensible. So that's my tuppence halfpenny work for how we can all try to do this better and constantly try and improve the way in which we communicate.